Hello, good morning, friends. My name is Dr. Amin, those who do not know me. So, we are here in the lovely professional university. Today, I'm going to demonstrate to you about the bacterial conjugation experiment. Okay, so rest of the steps you will see in the laminar hood, as you can see here. Okay, so let me bring it little bit close to you. So as you can see here, we have plates. Okay, so this plate is basically our donor plate. If I bring close to you, so the donor bacterial streak plates, this is in tetracycline resistant. Okay, the second thing which we need here is the recipient strain. So this is our recipient strain. This is streptomycin resistant. Okay, so two bacterial strain which we need. We are basically following the GenA kit. Okay, so which is also present with me. Now let me first sterilize my hand. I have already sterilized by UV treatment the whole area, so I will be showing you. Okay, so this is our kit. So the kit comes, this is a transduction and the what is called as your conjugation kit. So with the components I'm just showing to you right now. So just taking out. So as you can see, this one, okay, so this one is the LB broth. Okay, so the kit provides the LB broth. The second thing kit provide is the agar. The agar is required to make those plate, usually 1.5 to 2% of the agar which is required. Besides this, the kit also provide us the antibiotic selection. Okay, like here they have this is a transduction kit which I am showing. So they have, for the transduction they use a chloramphenicol and the ampicillin, which is in another video I will show. But we have the tetracycline and the streptomycin with us. So that's what I'm going to demonstrate to you. So that is comes with your kit itself. Then this is your calcium chloride they have provided for chemical transformation, which is another video which I have already shared. So this is required if one has to do a transformation. And these are the ampoules. So as you can see, this is the recipient strain. So this comes as a lyophilized powder. So we can basically we have to break this glass, and after breaking this glass, we basically grow into the culture. Okay, so like this in small tubes, or like in case of this falcon tubes which has been shown, 50 ml can be grown. So we have already done that thing for purpose. Then this is shown here is the donor strain. Okay, so the donor strain which has been shown here. Okay, so there is one another which I will demonstrate in another video. So these are the materials which we required. So what are the materials which you required? You required, first of all, the number one is that you required the strain. Okay, and the second thing which you required, the first thing is required, you need the strain. Then you need a autoclaved LB broth medium with suitable antibiotic. For conjugation experiments we required streptomycin and tetracycline. You probably need the appendrops, which is 1.5 ml tubes. Then you also need inoculating loops. Then you need the plates for doing the experiment. So this is about the bacterial conjugation experiment which I am going to demonstrate in my video number 2. First thing which is required in this video, I talk about what are the materials which is required for performing this experiment. Also I am telling you in briefly what happens in conjugation experiment, that the donor strain, they have certain plasmids. Okay, which is also called as the high frequency recombination plasmids, or F plasmids. and the recipient strain, they have the resistance gene for, in our case, is like streptomycin resistant, but they do not have a tetracycline resistant gene. So the donor strain has a plasmid or a high frequency recombination, either way can work. And they also have in some cases the helper plasmids also. So this allow the pilus to form, or the pili in plural. Now through this pilus, the donor strain will transfer one of its strand, that is if it is a plasmid bond or it is a genome integration in high frequency recombination, to the recipient strain. So if you allow the strains to do these conjugations for 5 minutes, 10 minutes and so, then it will transfer a resistance gene, making the recipient strains both, since you can say the resistance to streptomycin and tetracycline, which I am going to demonstrate in the next video. So stay tuned and watch that video. Thank you.